They are part of a large set that I pull from and will be presenting on in a few webinars coming up. So by all means, if you guys want to do that, I don't mind if you share on social media. And you could tag me also on Vitamin PhD. So tag me, give me a follow if you like this information. I have a lot more where that came from. So without further ado, I will bring up my little presentation and I will go ahead and share my screen. And then we'll get started. And yeah, if you guys have any questions, feel free to put them in the chat box. Or if you have, oh, I don't see like a hand raising thing here, but you know, by all means, or Mike, if you have a hand raising thing. So
if I see anything, I'll let you know.
Okay, cool. Yeah. So don't be shy. And if you guys like want to, you know, have face-to-face chat stuff and whatnot at the end, I'm totally down for that too. I do telehealth for a living, and I actually really love to be able to communicate with folks, even though we're many miles apart. Oh, thank you for sharing. Cool. Yep. So that's my website. You can see like lots of podcasts there, articles, links to my other website, which is all about coaching. So I run Bridging the Gap with Comprehensive Coaching with my good friend and colleague, Shannon Beer. So you can see the two hats that I wear. So here I go. Let me let's see if I can get this presented.
It's okay. They're used to me messing up all the time. So
okay, great.
Bar is very low.
I've switched over to using Canva because it's really nice that I can kind of pull from that to do like Instagram stuff and then also have presentations. But it's just a little bit weird the way that they have it set up to like when it presents, it like goes to full screen and you can't stop it. All right, so there we go. So I'm gonna share my screen and then let's see if I can get this to full. There we go. All right, yay. So everyone can see this, I hope. Mike, you can see it. All right, cool. So like I said, so giving you guys some updates and whatnot, kind of a brief overview of gut microbiome science. So this is probably what you guys might be accustomed to seeing. So if you guys are on Instagram or Facebook, anywhere in sort of the social media sphere, people tend to assume that gut health is one thing. It's one variable that we can modify, and it leads directly to the presence of health or disease in humans. And so this is where a lot of sort of the myths, misconceptions and confusion can come from, because we take a term that is sort of nebulous, gut health, and doesn't have a clear meaning, and then we extrapolate from studies that can't really get us there and come up with some causal relationship between health or disease. And it's understandable that we want to do this because we want to find interventions. I think one of the biggest complaints that I might get from presentations is, you know, what am I supposed to do about it? And the answer is, well, we don't know yet. Because the reality is that gut health is not a clearly defined entity. What we're actually looking at is the microbiome, and I'll give you the definition for that in just a second, but it's a collection of tens of trillions of organisms that are all interacting with each other, and then they may also interact with our genes. What you see there is SNP, that's single nucleotide polymorphism. It's basically one mutation in a gene. So our specific set of genes interacts with the microbiome, and then the microbiome may have an effect on a specific health outcome, but there are also going to be other confounding factors like our location, whether we were born vaginally, whether we were breastfed, medications that we've taken, whether we have a dog, our diet, our physical activity habits. All of those can influence the microbiome directly and indirectly, and obviously also can affect health outcomes. So this was from a really interesting paper in which they're trying to determine ways to study the microbiome that may give us a better sense of a causal relationship, or at least the directionality, because that's just one scenario. As you can see, there are three other scenarios, some of which are looking just at the role of our genetics on the microbiome, some of which have switch the directionality of things. So this is where we really are right now, that the best we can do is draw correlations. We can say, well, it looks like in people who have this disease, sometimes they don't tend to have this many of this specific organism. Does that mean anything? We still don't know. So the science is really in its infancy, And it's important for us to wait for it to catch up before we start trying to come up with interventions. Oh, doesn't want to click. There we go. So what happens when we put the cart before the horse and we don't let science really inform our recommendations? We end up with a lot of fads and fallacies. And what's really interesting is that we can see that there are many characteristics of fad diets, and this is something that was characterized by the Pennington Biomedical Research Institute about 10 years ago. They characterized a number of different traits as being a characteristic of fad diets. So things like promising quick results, making unfounded claims about certain foods offering specific advantages, of course, for things like weight loss or detoxification. There are foods that are against the rules or harmful or inflammatory, and then there are foods that absolutely must be eaten, things like requiring pills or supplements. We can see that this manifests in the area of gut health on Instagram and in social media with things like gut reset programs, or coming up with the magical kind of medicinal foods, you know, acai berries. Yeah, exactly. Like acai berries, celery juice, coconut oil. I'm trying to think of others, but it's like these foods that have these like magical properties. Or foods that are inflammatory, that, you know, kill the gut microbiome. And it's kind of funny, depending on, yeah, exactly, grass-fed butter. And it's like, depending on who you ask, something like meat should never be eaten or should only be eaten. I don't know if you guys have heard of like the carnivore diet, but, you know, like there are many people, even physicians, who think that vegetables are problematic and should be completely excluded from the diet. So we end up in these places of extremes, and we play this game of sort of like I don't know if you guys remember telephone, where you used to like whisper to the person next to you and they'd whisper to the person next to them. And by the time it got to the end of the row, it was a completely different message. And that's what we see happening when it comes to, you know, communicating gut microbiome science. We're taking something out of a petri dish, and then trying to extrapolate it to human beings. And then that's where we end up with like, oh, take collagen because it heals your gut. So I haven't, you know, I don't want to digress too far, but there are some real concerns about the potential effects on the people who are consuming this information, because there are certain beliefs and certain dietary practices that can actually predispose people to developing disordered eating or an eating disorder. And there are some characteristics like believing in food as medicine, in having very strict rules about foods that are off limits or allowed. Sometimes the practice of veganism for various reasons, or vegetarianism, that if people have strong beliefs about foods kind of being off limits, that could predispose them to some eating psychopathology, or the development of orthorexia nervosa, so sort of an obsession with healthy eating. So when we go to extremes, sometimes that can lead people to really develop eating psychopathologies and fear and anxiety around food. And even, you know, less extreme than that, they might just waste a lot of time and money. They might forego effective treatment because they're going for perhaps something that's an alternative treatment, or something that's completely ineffective, like doing a detox. So it's important that, you know, when we're providing information to people, that we're doing it in a way that empowers them. Now, this quote is from a recent article that I had pulled from that previous graphic that was talking about the problem of being able to establish causality. And they actually even mentioned some of these direct-to-consumer microbial analysis companies like Ubiome and Viome that are making some claims that you might remember from several years ago. They're pretty similar to the things like with precision genetic medicine. Like, we'll analyze your DNA and then we'll be able to tell you exactly what you should and shouldn't eat. They're doing the same thing with the gut microbiome, and we're really not able to do that. These microbial stool analyses are not giving us that type of information. And even if we took it directly from the intestinal passageway, from the lumen or from the mucus, we still wouldn't be able to give you that information. So they can just be very misleading. So it's very problematic in a number of ways. So what then is the gut microbiome? So the gut microbiome is different from gut health, although I think people might be using it synonymously. But gut health is really a nebulous term. It doesn't have a clearly defined meaning. So we want to talk first about what the gut microbiome actually is, and then we can talk about what people might mean when they say gut health. So the gut microbiome refers to all of the microbes, all of the microscopic organisms, and all of their genetic material that reside in your digestive tract. So this is going from your stomach to your anus. So one end to the other, all of the microbes in there. You actually also have an oral microbiome that's separate from the GI microbiome, and you have a skin microbiome, and females, you have a vaginal microbiome. So we are inhabited by tens of trillions of microbes just in our GI tract. It's really an impressive number of cells, and they weigh about a couple pounds. So you can just imagine like that amount of biomass. Now, back in the day, there was this kind of trope that went around that was like, oh, you're more bacterial cell than you are human cell. That's not entirely accurate. You're actually about a 1-to-1 if you take into consideration like all of your blood cells. But in terms of genetic material, they vastly outnumber us, which is super helpful because that means we can outsource some of our metabolic processes to these microbes. So they help with energy harvesting, they help with pathogen resistance, they have very likely a role in just our overall human development from the time that we're born all the way through elderly adulthood. They play a role in brain development, in immune development. They synthesize very trace amounts of some vitamins for us. What I mentioned, energy harvesting, they actually can take fibers, which are indigestible to us, and ferment them to short chain fatty acids, which are then available to us for energy. So they really are essential to our development and our ability to survive and thrive. And what we find in rodent models that are reared without any gut microbes, they call them germ-free mice, they're actually developmentally delayed in every way. So neurologically, immune, they are resistant to weight gain. So really, it's very clear that the microbes have a symbiotic relationship with us in many ways, in that we provide to them a safe place. So we give them the real estate, and we hopefully are giving them plenty of microbe accessible carbohydrates from our diet. And in turn, they produce nutrients for us, and they fight off pathogens. Now, of course, we don't have a fantastic relationship with all of them. There are some opportunistic pathogens that reside in our gastrointestinal tract as well. But it's just like the force, we need a balance of good and evil. We actually do need some of those potential pathogens. And the potentially pathogenic microbes don't always cause disease. They actually also play a very important role in informing our immune system. And as long as they are in relative balance with the neutral and beneficial microbes, they won't express their virulence factors. So they won't actually produce the things that can make us sick. So just the presence of a potential pathogen doesn't necessarily mean that we have an infection or an illness. And of course, we have plenty of those beneficial microbes as well. Those are the ones that are producing what we call postbiotics, or products that are beneficial to us in some way. Now, it's not just bacteria, even though we talk about them a lot. It's about 99% bacteria, and most of them are located in our colon. But we are also inhabited by fungi, archaea, and even viruses that can infect both human and bacterial cells. And so these microbes are all interacting with one another, and they're interacting with our intestinal cells, and they're interacting with our immune cells, and they're also indirectly interacting with our peripheral tissues. So you can see that the gut microbiome is not one entity. It's a collection of trillions of living organisms interacting with one another, and what you could consider to be sort of an entire ecosystem. And when we look at the various regions along the intestinal tract, when we're looking at the small intestine versus the large intestine, or even when we're looking in the lumen versus in the mucus near the cells, there are little microhabitats that are enriched with different ratios of these microbes. So when we use a term like gut health to refer to something like this, we're really losing so much of the resolution and the detail that we need to keep in mind. So gut health, I think when people are talking about gut health, they're probably talking about, the lack of a disease of their digestive tract, and they are having comfortable bowel movements and maybe like not experiencing gas and bloating and things like that. But I think that some people might assume that there's a problem in there just because now they know that it exists. And so they find that, now gut health is a thing. Gut health is a problem that I could have, or it's something that I could optimize, or gut health is a problem that we can sell to someone else so that we can sell them the solution afterwards. Now, that being said, there are diseases of the gastrointestinal tract. So we have functional diseases, so diseases sort of the physiology. So there's nothing wrong that we can see with the structures there, but they're not working quite like they should. And then we also have diseases of the actual tissues of the GI tract. Now, again, we don't have a causal relationship between the gut microbiome and any disease. And we don't have a directionality with it either. So even though we do see some correlations in certain disease states and certain microbes, we can't say that one causes the other, because it could be that the abnormalities of the GI affect the microbes, or vice versa, or it could be a two-way street. It doesn't have to necessarily be unilateral. And there are some indications that the microbiome may play a role in peripheral diseases without any changes or any known disease in the GI tract. For example, they've been implicated in the development of metabolic dysregulation and type 2 diabetes. Some folks might experience digestive discomfort. So they might feel some gas and some bloating, and then perhaps assume that they have, bad gut health, or there's something wrong with their microbiota. But in reality, those microbes as living organisms have to metabolize nutrients, and byproducts of that metabolic process include gas and short-chain fatty acids. Certainly, there are some potentially problematic compounds that have been associated with colorectal cancer, but just the production of gas doesn't necessarily indicate bad gut health. It's just that those microbes are alive and they're doing what they would normally do, and we have to pass that gas, or sometimes it gets trapped and then we have some discomfort. So quite often, foods that are sort of what I call, good for the gut, so foods that contain those microbe accessible carbohydrates, can lead to gas formation and can lead to loose stools because they're osmotically active. They pull water into the gut. So it might feel like they're not good for gut health. They might make us feel poorly, but that doesn't necessarily mean that they're bad for the gut or that they're damaging our gut. It's just that they are being fermented, and that leads to the production of gas. But I think you can see where some of these things get conflated and confused, and people are now more concerned about the idea of gut health because it's something that they're more aware of. Now, I want to cover a couple key terms that are important for understanding gut microbiome science and what people might be talking about. So one of the big words that we use when we're talking about gut health is diversity. So diversity can be measured in a number of different ways, and it doesn't always mean the same thing. But generally speaking, when we say diversity, we're talking about the richness and the evenness of the microbial population in our GI tract, as well as the variety of genes. So we can look at this in terms of taxonomy. So that's who's there. It's the number of species, that's richness. Evenness is the relative abundance or the representation of each of the different groups. And then we can also look at phylogeny. So how related are they? And then when we look at functional diversity, we're looking at what are they actually doing. So we might be looking at, these different omics approaches are looking at the metabolites that are being produced, looking at the proteins that are being produced, and looking at the genes that are present and the genes that are actually being expressed. So when we look at all the genes that are available, it's like looking in a book of recipes. Doesn't necessarily mean that all of the recipes are being made or that all of the genes are being expressed. We're just looking at sort of the functional capacity of the gut. Now, these actually tell us two very different things. What's really interesting is that there seems to be sort of a functional redundancy in the human microbiota. That means that even if we lose some of who's there, others will pick up the slack. So it's not so much, so the importance might not be really in who's there and looking at like the taxonomic diversity. The importance may lie more in what are they doing and what can they do. when we look at the loss of diversity, we might be looking at a loss of who's there, so fewer species or less even distribution. It's less common for people to be looking at functional diversity. So to say like, well, okay, so we lost some of the taxa there, but what can the gut still do? What can the gut microbiome still do for us? And what they found in recent years, as the technology has improved, is that individuals who have obesity, for example, have lower functional diversity, even if the taxonomy doesn't differ very much from people with lower body mass. And that in certain disease states, they're looking more at taxonomic diversity, and that there may be an overabundance of pathogens or a loss of beneficial species, but we're still not quite clear on like the functional changes that are going on there. So ideally, as the science improves, we are going to be looking at both of these more often. And the reason that we haven't done that till recently is just that it's really expensive. So like whole genome metagenomic, whole metagenomic shotgun sequencing is like a long way of saying, yeah, we're looking at like all of the genetic material that's there. Because sort of the classic ways we started out, it was just culturing. So we like swab something, see what grows in a petri dish. That was super limited. Then we were able to look at the DNA or the RNA, but that has limitations as well. So the more expensive it is, the better it is, but then, you know, the harder it is to get funding to do that. So the other word that we use a lot is dysbiosis. This is another word that people use synonymously, especially in social media, to refer to sort of bad gut health is what that's how they use it. Now, dysbiosis is actually a term from the literature, so it's not something that was like just made up. But again, it's sort of nebulous. It doesn't have a clear definition. It really just means any alteration compared to a control group in a study. And as you can see, it has in the literature been used in many different ways. And researchers aren't always clarifying, you know, their definition of dysbiosis. And dysbiosis has become sort of a boogeyman, sort of a scapegoat. This is another great quote from an article. This is more of an editorial written a few years ago. But I love that the article's title was "Dysbiosis is not an answer." And he basically goes on to say that because it's such an ambiguous definition, we can kind of use it when we don't know what's going on. Like, this is our explanation when we don't have a better explanation. And now people are using it as sort of a diseased state. They're saying like, oh, you have dysbiosis. You know, that's a problem. And clearly dysbiosis can cause absolutely anything because no one really knows what it is. So if you have headaches, fatigue, acne, irritability, trouble losing weight, trouble gaining weight, gas, bloating, really anything, people will be like, oh, it's dysbiosis, you have bad gut health. And guess what? This sells really well because people can say, oh, I'll help you correct your dysbiosis. And because it's a made up thing, there's no way to say that you don't have it. And then there's no way to say that they didn't fix it. So wow, money making. So you know, you're welcome, you guys. This is your next big thing. Now, that being said, there are groups that have gone through the literature, thank goodness, and tried to identify patterns in microbial populations and people with varying disease states. Now, when we look at two people, even with the same disease state, the comparison of those two people, their microbial profiles will be incredibly different. So much so that we wouldn't be able to find really much in common at all outside of sort of the core human microbiome. So about, you know, 1/3 of our microbes are just human, and then the other 2/3 are like very individual to you. But when we look at huge populations over multiple studies, then we might be able to identify some patterns. We have done that, but the patterns are still really general. So what you're looking at here on the left-hand side is from a meta-analysis a few years ago, where they tried to characterize dysbiosis. And they were kind of thinking, maybe we'll find one dysbiosis, but they didn't. They found multiple dysbioses, either characterized by an enrichment of pathogens or a loss of beneficial microbes. But in some cases, there wasn't a clear pattern. And so that's very likely due to, the heterogeneity in how we're actually measuring who's there, but also just inter-individual variability is so significant, and it's such a limitation. The other thing that we have to keep in mind is that the vast majority of these studies are using fecal microbiome samples. The fecal microbiome, what you'll see in your stool, is actually significantly different from what you'd see throughout the rest of the gastrointestinal tract because of those microhabitats that I had mentioned earlier. So when we're looking at stool samples, it's quite representative of what we would see in sort of the distal section of the colon, but it really misses out on what we might be seeing throughout the rest of the GI tract, or even sort of cross-sectionally in the lumen versus the mucus. So when we use terms like diversity, when we use terms like dysbiosis, know that these are still fairly nebulous terms, not as nebulous as gut health though. So just keeping that in mind when we're trying to differentiate between like, oh, what is good gut health versus, what is bad gut health. So to give you guys some practical implementations in terms of navigating the literature and navigating content that you might see on social media, there are some sort of gut health red flags, verbiage and phrases and practices that you might see from one of my clients. I love this term. She called them like gut health journalists. I don't want to like poo-poo on anyone's intentions or content. I'm sure that like most people do want to help those who are consuming their content. The problem is just that even within like the microbiome research world, even other researchers are like, why does the data look so confusing? Like, why do you make these graphs that are beautiful but illegible? And like, what do you mean when you say dysbiosis? It might be different from what I say. Can we have some like clinical cutoffs or something? We just don't have a lot of clinically relevant, applicable data at this point. So if you see some of these terms, just be very cautious about maybe sharing or consuming that person's content, especially if they're using if they're using terms like toxins as a single entity and then promoting maybe a detox or a gut reset. It's just not, a gut reset is not a physiological possibility. Claims about healing the gut. So certainly tissues within the gastrointestinal tract can be healed. There can be the presence of a disease and then that can heal. But when people say just like healing the gut, that again is very nebulous. Are we talking about like the entire length of the gastrointestinal tract? Are we talking about having an effect on the microbiome and so on? Some extreme elimination diets. So I mentioned the carnivore diet earlier. So that's one that completely excludes all plant matter. It's only animal flesh and organ meat. And there's it's not intended as an elimination diet. It is intended as a long-term diet or any kind of specific diet dogma. Really any dietary pattern can be done in a prudent way. Even if someone wanted to do the carnivore diet for a short period of time as an elimination diet with a reintroduction, I wouldn't completely poo-poo on that. But when it becomes dogmatic and there's no evidence to support, that it's a health promoting dietary pattern, there's no evidence to support the claims that they're making, that's just dogma. That's just someone making things up and having a strong opinion. Any statements about the gut microbiome causing other things. There are known pathogenic infections. If you get listeria, if you get certain strains of E. coli, if you get Giardia, yes, you will have severe gastrointestinal distress, and that is an infection that requires treatment. But when we say that the whole microbiome causes any specific disease, that has not been shown in the literature at this point. We just are not able to create, to extrapolate a causative relationship. And then when people, extrapolate to say that things might cause, that the gut might cause things like, mental fog or fatigue, these are things that don't have a known etiology, they don't have a known cause. And so that makes them, ripe for saying, oh, it's dysbiosis. Or if people are, saying things like good gut health or bad gut health without really trying to explain like what it is they mean by that. I have had so many discussions with other practitioners who, also have experience or education in the gut microbiome literature. And we're like, what else can we say besides gut health that people know what we're talking about? Like, I don't want to say gut health. This is a problematic term, but it's the one that most people are familiar with. So we have to kind of use it, have to meet people where they are, but we also have to explain what it is we mean by that. And so I tend to try and say things like gut health science or gut health and the gut microbiome, or at least give a working definition like I did today. Now, as you are interpreting the literature, there are some key questions that you might want to ask yourself. Now, this article was from Patrice Connie. He's one of the, I think, like godfathers of microbiome science. He actually coined the term metabolic endotoxemia, which refers to low-grade inflammation as a result of some toxins that can exit the gut. He was the first to really see, oh, hey, high-fat feeding seems to potentially be problematic. But even he has emphasized that we are potentially drawing causal relationships out of data that can't provide them, because there are so many other factors that need to be considered. So when you're reading really any paper, you can ask yourself things like, do the detected differences actually matter? So when we're looking at tens of trillions of microbes and we're looking at hundreds of species and potentially thousands of subspecies, and we see a change in one group, does that actually matter? Well, we don't really know, because we don't know what is that relative to the magnitude of the entire microbiome, and also what's the clinical outcome? Does it really matter at all, even if there's a significant change? Then of course, does this study show a correlation or establish potential causation? Now, when we use rodent models, that gives us really excellent mechanistic data, and we can start to tease out a potential causal relationship. The way that we do that is we take a fecal transplant. So we take a stool sample from a human donor, and we transplant it into a germ-free mouse. Now, there are a couple of limitations with this. One thing that's kind of a sneaky secret, I think, that people aren't talking about or might not be aware of, is pseudo-replication. Now, this might be an ethical dilemma, but if you take one person's fecal sample and you transplant it into 5 mice, now is that an N of 1 or an N of 5? Because you've taken it just from one person. Well, in the microbiome literature, that's an N of 5. So, things like that to keep in mind. Also, when we do that transplant, it does not come out in the mouse looking exactly like a human microbiome. It is a mouse-flavored human microbiome, because the mice have a very specific anatomy to their GI tract. They have a different diet. So we can't take rodent data and say, oh, yes, this is exactly what we see in humans. And that's really where we are, especially I know one person had asked about, the microbiome in the brain. Looking at the gut-brain connection, it's primarily rodent data. And I hesitate to even say that rodents have moods. I don't know. Does a mouse have a mood? Can a mouse feel sad? Are we really doing ourselves any favors by saying oh, the gut microbiome in rodents alters their mood? Another ethical, dilemma, I guess. But when we're looking at human data, when we're looking at just observational studies, for example, when we're looking at the effects of, alcohol intake on the gut, we don't have intervention studies yet. All we have are observational studies. So we look at groups of people who drink versus people who don't drink, and we say how do their microbes differ? And then looking at the potential mechanism of effect. So was this something that was really due to the intervention? Or could it have been that we affected one microbe and then it affected another microbe, and then we assumed that the intervention affected that secondary microbe directly? Then how well does this experiment represent reality? Are we looking at applying artificial sweeteners to a cell culture model of intestinal cells, or have people actually ingested the artificial sweetener? Because if you want to look at the magnitude of effect of artificial sweeteners and you want it to be really significant, put it on some cells or feed it to a mouse, because if you put it into a human, you're really not going to see a huge result. And then, could anything else explain the results? So what else is going on? Was there a dietary control? When we modified the diet for that intervention, what other, if we change protein, other macronutrients change as well. Was there a change in fiber? Did they have controlled physical activity levels? There's so many things that influence the shape and the activity of the microbiome that even when we're looking at just one intervention, very likely, we're not able to control to that extent to make sure that it's nothing else that has played a role. So just those things to sort of keep in mind as you are navigating the literature and, you know, getting a lot of potentially conflicting and confusing information from those around you. And as people are asking you questions, because, you know, going into the field and being professionals, people will ask you about the latest hot topics. So without further ado, I can, excuse me, I can answer the questions that came in ahead of time, and then I can also answer any questions that you guys have that have come up in the last little bit that I've been talking, or unless we want to kind of do it like all at once. What do you think? And like intersperse? Or if you have any preference?
Since they're being quiet, it usually takes them a few moments to talk. I'll throw out there that, you know, feel free to go ahead and touch on the subjects that they, you know, the questions they had. And though, yes, thank you for bringing up the key difference between humans and animals. It's kind of like looking at the old cholesterol research where initially it was feeding diets to vegetarian animals that were heavily meat-based, and you're wondering why a rabbit freaks out because it's a little bit different there.
Yes, yes.
But the other component that I personally just think it'd be fascinating, because you've already touched on it, which was specifically when you're talking about cell line research and the effects on nutrition. We haven't really covered that too much yet in the course.
Okay.
So if you want to give them a little bit more context on how obviously there's some good initial information there, but obviously extrapolation gets dangerous pretty quick.
Yes, yeah, absolutely. So when we're looking at the effects of, I'll bring up artificial sweeteners because this is the one that I think most people are concerned about. The studies that have been done in cell culture, we call that in vitro. They will do it in a couple different ways. So sometimes they may grow intestinal cells in a dish, and those will be living human intestinal cells. And then they will apply media, so they'll apply cell media that they would normally kind of exist in, and then they will give some concentration of an artificial sweetener, and they might titrate that up, so they increase and increase and increase the dose, and then they measure maybe cytokines, so like inflammatory chemicals that cells will release in times of like inflammation to fight off a pathogen or to like start initiate an immune response. So they'll measure levels of those chemicals, for example, or they might measure cell death. So they might say like how many of these cells survive. The other thing that they'll sometimes do is they will take a few representative groups of bacteria. So we call them Keystone Species. So there are some bacteria that we would expect to find in a human gastrointestinal tract, things like F. prausnitzii, Akkermansia muciniphila, a bunch of different bifidos, E. coli. So we take those guys and we say like, okay, we're going to put a bunch of these in an artificial gastrointestinal tract and kind of let them hang out and grow together until they become like a stable microbiome. And then we're going to then we're going to dose them with artificial sweeteners. And again, we're going to titrate that dose. And then we're going to see what they do. Like, do they die? That's what they usually are looking for. How do their growth rates change? Do they metabolize these at all? Like, what happens to them? But that is neither one of those are really physiologically relevant. Cell culture studies are really helpful for being able to look at signaling pathways. So this is something that I had used. We used to take skeletal muscle samples from humans. We would culture them in dishes. It was really cool because especially the cardiac cells will actually contract together. So it's really interesting to watch that. Like they just have, like their own little pacemaker mechanisms. But so you take these cells, you put them in a petri dish, you add a bunch of growth media, and then they differentiate and they act like muscle cells. So they express the genes that are characteristic for skeletal muscle cells. And then we would dose them with varying concentrations of glucose or palmitate or LPS, lipopolysaccharide. That's the endotoxin that comes from the gut, that metabolic endotoxemia. And then we would measure how they would metabolize those. And from there, we could say like, oh, these skeletal muscle cells, after we dose them with LPS, they don't break down fats so well. That's interesting. now we should look at this in actual rodents. Okay, that's interesting. Now we have to look at how this actually manifests in humans. So you start with cells to give you the idea of the potential mechanism of effect. And then you go to a rodent model because they're less expensive. You can kill them when you're done with them. And it's easier to collect their tissues. But again, you're looking more at mechanistic data, but now it's in like an actual, multi-celled organism. And then you can move on to something more advanced. When it comes to the human gut microbiome, the closest proximity I would say would be, looking at like porcine, so like pig models is one thing that they're looking at expanding into, or other primates. But they really have to find living creatures that are omnivorous, that have a GI tract that is similar in structure to ours, because the diet the gastrointestinal tract and the diet are closely related. So like carnivores have a very simple digestive tract, whereas like an obligate, like an herbivore, like full herbivore cows, they've got like 4 stomachs, so that's not going to be a great model for us. So yeah, that's where it gets really tricky, because we can give cells an amount of artificial sweetener or rodents that like humans would not be able to ingest. So the cardiac muscle fibers, we took those. Well, we would like sometimes take them from the mice, or you can actually buy them. You can just like get them, you can order them. So they send to you like little cardiomyocytes, so like little like baby cardiac cells, and then you put them in a dish, and like as long as you put all the like growth factors on them, they'll differentiate, or they die. I mean, so cells die all the time from nothing, which is really troubling. So yeah, dosing them with a bunch of nonsense, they will die for sure.
It's mildly terrifying, because I've done muscle biopsies and seen a little bit of tendon biopsies, and those were always on humans for the lab I was at Kansas. So I'm like, wait, cardiac fibers? Like,
yeah. Oh, no.
That mouse is not walking that off.
No. Yeah. That's the benefit, I guess, of animal research, is that you can collect any tissue you want afterwards. So yeah, I think the other questions one person, I really thought that the one question that the person had asked about sort of remodeling the microbiome in a way that lasts, or modifying it in a lasting way. But the gut microbiome is actually, it's very resilient, which is a good thing. We wouldn't want our microbiome to completely we wouldn't want it to be like flaky. we wouldn't want it to be flippant. It's good that there's functional redundancy there in the genes that are available and the genes that are being expressed. And okay, reconfigure, that's what they said. So there's redundancy there. Now, we haven't looked as much into the functional stability, but in terms of taxonomic stability of who's there, current estimates are that about 60 to 80% of your microbes hang out with you no matter what. You basically, like, you're colonized pretty much at birth. It's very dynamic for the first few years. You've got, there are changes in the anatomical structure of the GI tract. It's much more permeable when you're born, so there's more oxygen in it. It becomes less permeable and more anaerobic as you age. Then you've got the inclusion of dietary carbohydrates, and so that gives rise to blooms of many other microbes. So you really see huge increases in diversity when you introduce solid foods. Things stabilize at around age 3 or so, and then throughout adulthood until you get into about your 60s, it's fairly stable. So you may see some changes with lifestyle changes. So they've looked at exercise interventions and dietary interventions do cause changes rapidly. So within about 24 hours of a dietary change, you'll be able to measure changes in who's there. And even in less time than that, you'll be able to measure changes in functional capacity. Even they've looked at like before and after marathons. you see a change in what the microbes are actually doing. I kind of like to joke that like they had some increases in like flagellar gene expression, and I'm like, oh, they're trying to like hold on with their little flagella because you're jostling them all around. That's not actually probably true. I just think that it's really funny. I thought that was a neat change. I'm sorry if you guys are hearing dogs in the background. Is that super loud? My dogs are like going crazy right now. Hopefully not. Is it okay? Oh, I would if I could, but they're like all in different areas. We've got an Australian Shepherd who's like 8 months old, and then I have a English Bulldog mix who's about 12, and then a Jack Russell Beagle who's also about 12. And then we also have 2 cats, so it's like a full house in here. Fortunately, the cats are quiet, but I can like send you some pictures afterwards or something so you guys can see them. But I'm like locked in the office right now. But I digress. So the microbiome is very stable. And even when we see those acute changes, like after an exercise bout or, the 24 hours within a dietary intervention, the magnitude of those changes versus like the overall stability of the gut, probably it's still really stable. You're not really able to completely reconfigure. And what's interesting too is that even when folks do an intervention, like say they do a dietary intervention or they start exercising or something, obviously when they stop doing that, the gut microbiome actually reverts to baseline, but sometimes it actually reverts closer to baseline even when they keep those changes up. So the gut microbiome is, it's dynamic, it's constantly adapting to challenges, but it is also very stable in that we don't see huge losses or huge balloons. That being said, sometimes that does happen in the case with, you taking a strong antibiotic and that gives rise to a C. difficile infection. That, yeah, sometimes we can see that these opportunistic pathogens, when they get the chance and they start to grow for some reason, they might grow out of control. Or in individuals who are taking proton pump inhibitors or calcium channel blockers, some various, I think drugs for controlling acidity, that changes the pH in the small intestine, and then that can give rise to abnormal growths of bacteria that wouldn't normally be there. So we can see some fluctuations, we can see some abnormalities, but on the whole, you're not ever going to be able to completely clean everything out, redo it in a better way. Like that whole idea of a gut reset is just not physiologically plausible. We don't even know which microbes like which type of fiber. Like we can't even get it to that point to be like really, tuned in. It would be great if we could, because then we could say, instead of taking like a broad spectrum antibiotic that might knock a bunch of taxa out, we could just say, oh, just take this one thing. This will just get only the pathogens. We're not there yet. We're not able to do that. So that's why that's a red flag when someone's like, oh, we can just get rid of the bad guys and then like only have the good guys. Even considerations about who might be quote unquote bad and good, it's very contextual. Lactobacillus is one that is a butyrate producer, so it helps with energy harvesting. And I've seen some physicians say, oh, Lactobacillus, that's a bad guy, he's associated with obesity. Well, A, no, not really. And B, if you are a person who needs to harvest more energy from the diet, that could be very beneficial, especially if you're in a developing country and your energy availability is very low. So keeping those things in mind. Now, if you want to engage in practices that seem to be evidence-based in supporting a diverse and an adaptive microbiome, it would be taking in a variety of dietary fibers from fruits, vegetables, whole grains, legumes, and nuts, because we don't know who wants what type of fiber, so just give them a whole bunch. And then to engage in physical activity. So not so much that you're, having like suppressing your immune system, but regular physical activity is also associated with increased microbial diversity. Can the gut microbiome have significant effects on brain function? Likely, yes. But again, we're looking mostly at rodent models. It probably plays a significant role in early brain development. And it does seem to play a role or have a relationship with eating behaviors and appetite, both potentially through the production or modification of neurotransmitters that might regulate appetite, and also through the production of some of those postbiotics, those short chain fatty acids such as butyrate that may also regulate appetite. There is ongoing but still early research looking at the role of the microbiome in things like anxiety, depression, eating disorders, and autism, but we don't have, again, really strong evidence to point at what could really be going on. There have been a couple studies looking at fecal microbiome transplant. This is something, they found that fecal microbiome transplant from mouse to mouse seems to have an effect on their mouse personality, or from individuals with a psychiatric disorder into a mouse also seem to affect mouse behavior, is what I would like to say. And so they have done some fecal microbiome transplants in individuals with autism, but they didn't have a control group. And so we don't know whether that the improvement in their subjective experiences was due to the transplant or due to just aging and the passage of time. In studies looking at the effects of probiotic supplementation on psychiatric disorders, there's again not strong conclusive evidence, because it's about, half of folks seem to improve and the other half don't. There have been some interesting, really small studies looking at, certain foods and mood. But again, it's like, my answer to you is yes. I'm sure yes, but we just don't know how. And then alcohol and caffeine. This is really interesting. So I don't know if you guys have heard about like the whole 30 plants per week sort of meme thing that's going on. So that came from the American Gut Project. So back in the early 2000s, Rob Knight out of UC Davis, I want to say, he and others worked together to put on the Human Microbiome Project. I say put on to do this huge collection of human microbiomes from a multitude of different human body sites, and then to characterize them. And that had two phases. One was just sort of like, let's look at all of the microbiomes. And then the second phase was like, let's look at specific things like pregnancy. And then the spinoff of that has been the American Gut Project, where you can pay $99 and send in your stool sample, and then they will sequence that. So they'll look at the microbes present, and then they put your data in with everyone else's. It's pretty cool. You get to see like where you are in comparison to other people in the United States. And they also collect questionnaires. So they want to know like about people's dietary practices, their alcohol intake, their physical activity levels. And that's where we got the number 30 plants per week, because the people who reported eating at least 30 plants per week had higher microbial diversity than others. But what's not reported there is that also people who reported using antibiotics recently also had increased microbial diversity, but no one's like, take antibiotics, because it was probably, some of these are just like strange artifacts, just strange findings that don't seem to make sense. So alcohol intake went the same way, that the people who reported moderate alcohol intake, so they had, alcohol, beer or wine a couple times a week, also showed increased microbial diversity. Now, it's not because alcohol is fermented, we're not getting like live microbes from the alcohol, but it could be the polyphenols in the red wine that are metabolized by microbes that could play a role. But they're not really sure. And then with caffeine, they've looked at coffee and found that coffee also might be slightly bifidogenic, so it increases numbers of bifidobacteria and also is correlated with increased microbial diversity. Yeah, that could be it. So you guys, you better pick your partners well and make sure that they have good microbiomes, because you do exchange microbes from person to person. So that's another thing. Okay, so let's see. This is a cool question. Okay, so what type of changes do you see in gut microbiome when someone has a sudden radical diet change, like going from a normal balanced diet to carnivore or ketogenic or vegetarian? How quickly do those changes occur, and what outward physical signs of those changes can be seen? We don't have, oh, that's a good question too. So we don't have one specific profile of like, this is a vegan microbiome, and this is a vegetarian, and this is like a carnivore microbiome. We've looked, and people's responses to the diet are in part determined by their baseline microbiome profile. So like who you started out with determines who you will end up with after you've done some sort of dietary intervention. But one of the landmark studies out of Turnbaugh's lab, he's the first one who transplanted fecal samples into rodents and found that like, oh, if you transplant fecal samples from an obese person into a rodent, that rodent then becomes obese, even without an increase in food intake, potentially because of changes in energy harvesting. So that lab has really looked at at the changes in response to dietary changes. If you completely remove dietary fiber, or if you're very fiber deficient, you will see reproducible losses of bifidobacteria. That's one beneficial genus of bacteria. You'll also usually see an increase in Bacteroides, not to say that they're bad guys, but they do produce some compounds that are associated with colorectal cancer. So they're fermenting more of the amino acids. And when we ferment amino acids from dietary proteins, that's when we tend to see more of those potentially harmful compounds as opposed to fermentation of dietary carbohydrate. And likewise, if a person were to go in the other direction and say go from a fiber deficient diet to one that does contain adequate fiber, they would see an increase in bifidobacteria and generally speaking, an increase in microbial diversity and sometimes an increase in those short chain fatty acids. Those changes can occur within 24 hours. The outward physical signs of those changes probably would not be seen. You might see a change in the person's stool quality. You might see a change in their gas production. You may see a change in their body weight because of changes in energy balance, but you wouldn't necessarily see outward changes you wouldn't see an outward representation of the changes of the microbial community, if that makes sense. So that's part of the reason that it can be confusing and maybe troubling when we're trying to help people in this area, because they're like, I don't feel good, that must mean that something is wrong, or I feel fine and that means everything's okay. Those are not actually accurate statements. So increased inflammation. Inflammation is another word that we have to be kind of careful, and I'm like guilty of using it, without very clear definitions. So when we're talking about the inflammation associated with that metabolic endotoxemia, it is theoretically, because it's not like a proven thing that happens, theoretically it is systemic. So what happens in that situation is that there are certain types of bacteria that have in their cell wall this toxin called lipopolysaccharide. And when they die and they break down, the lipopolysaccharide is free to leave the intestine. So it can theoretically kind of leak out between the intestinal cells, potentially due to increased intestinal permeability. Some people call that leaky gut, and that's another term that's been kind of like co-opted and abused, but intestinal permeability does actually occur. When the LPS goes out into circulation, it can bind to certain immune receptors that we have on skeletal muscle and pretty much actually everywhere. But when it happens, that causes the release of inflammatory cytokines. So things that will activate our immune cells. And that inflammation is not the same thing as what we would see. You're not going to see like redness or things like that, or swelling. And it's not going to be like sepsis where you need to go to the hospital. It is below septic levels. There's actually not a clinical range for metabolic endotoxemia. So you wouldn't be able to go and like measure these and be like, you've got metabolic endotoxemia. The way that we do that in research is we just measure levels of LPS in the blood, but we actually don't have a clear-cut range for that either. So it's like, no one tell that we actually don't know what we're talking about when we say metabolic endotoxemia. So it could certainly occur sort of acutely and just in the digestive tract. So you could have the way your digestive tract is organized, you've got your intestinal cells, your absorptive intestinal cells, and you have this connective tissue supporting them. And within that, you've got a lot of immune cells. So they could certainly reach up into the lumen and come into contact with pathogens there and then initiate an immune response. Oh, it's so funny. I'm glad that you asked about kombucha. I know. Okay, so funny story for those of you who are super nerdy, and I probably no one else finds this funny, but if you're familiar with a systematic review, it is a secondary analysis of a body of literature. And sometimes they'll do a systematic review and a meta-analysis where they do like a secondary statistical analysis. And they're trying to determine, they're trying to sort of evaluate the quality of the evidence and see if they can form some sort of consensus based on what a bunch of similar articles on one topic might say. So a couple of years ago, a systematic review on kombucha was published, and it had one study in it. And you know that those authors were like, screw you guys and your stupid kombucha stuff. Like, where's the evidence? You know, like one study. And it wasn't even like a real, I think it was like something in Cells. So they were like, hey, there's actually no evidence for this. Did you guys know that? Like, okay, here's a systematic review, like up yours. A couple years later, another group also wrote a review on kombucha, and they cited that paper. And they also said, we went a step further. We went and looked at even the, can I swear? Okay.
Oh, yeah.
They went and looked even, we went and looked at even the shittiest of publications. Like, we have no standards. We're going to try and find any little piece of evidence to support what you're saying about kombucha in humans and like its magical curative properties. And they're like, hey, so we found this one article and he cites himself like 13 times. And then he cites this other article from like 1937. And then he cites some websites. So it was just hilarious to like go down this rabbit hole of kombucha. Yeah, because I'm like, this is something people ask about all the time. And so I'm writing a book for RP, I'm like finally freaking almost done with it. And so we're having a section on fermented foods. And I was like, oh shit, I've got to like add kombucha in here. And there's really like nothing that I can say in support of kombucha. And then the other thing is they were like, yeah, we found some case studies in humans, and actually like some people got really sick and one person died because, you know, they're making their own kombucha at home and they're not controlling the microbial growth. So Jacob, yes, potentially kombucha could lead to some negative effects if you make it incorrectly at home and then you die. But really, like the benefits on gut health, I mean, pretty much unfounded. Like they do, you know, they're basically saying, okay, tea has all of these potential beneficial compounds. It contains all, you know, like catechins, all these things. So probably fermented, then we're getting all of those compounds and maybe some live microbes. Then it has huge health benefits. And we've just like not waited for that to actually be supported by evidence. We're just like, yeah, it makes sense. Let's just say that that's what happens. So yeah, some people do live by it and drink it probably a little bit too much. Kombucha tastes really good, but just like with any fermented food, they're not like magical curative properties. We don't know which microbes we're actually getting or in what amounts. And the microbes that we use to ferment foods quite often are very specific to that food matrix. Like they want to live in yogurt, and then we put them into the GI tract and they are not going to necessarily thrive there. They don't necessarily have to exert an effect. So it could be that even non-viable or dead microbes pass through and interact with those that are there and then cause some change. But again, we just have to wait for, you know, like someone to do a study worth something and then, you know, see what happens. Because right now, all we really have, we do have some intervention studies in fermented foods, looking at like fermented dairy is the one that's studied most commonly. And that does seem to have some benefits in terms of like improving lipid profiles and like insulin handling in individuals with type 2 diabetes or metabolic syndrome. That seems to have like the most support, which is really funny because people who are like super gut health tend to also be like super anti-dairy. They're like, dairy is inflammatory. And I'm like, oh, it's actually one of the most evidence-based interventions. So, but stick with your kombucha, I guess. So yeah, so there's your answer to kombucha. Drink it if you like it, I guess. I don't know, make you gassy.
So one quick question, don't make it quick. You take as much time as you would like,
yeah.
Which is effectively, how far away do you think we really are research-wise before there's going to be some legit, clear indications of a better working definition of an optimal microbiome?
We're
not going to hold you to it, but
yeah.
is this a decade away? Is this 5 years away? Like, because I know a lot of people think we have solutions now, and we got an idea what's bad and what's good, but we don't have a clear here's the trajectory.
Right. Well, I mean, the Human Microbiome Project, I want to say that started back in 2007. It was either 2001 or 2007 because there were two iterations. I just can't remember which. But even so, it was really awesome. I actually got to meet Rob Knight back in March, like very early March. They had a Human Microbiome conference, and he talked about the Human Microbiome Project and the AGP and whatnot, and they have like so many terabytes of data. They have so much data, but they don't have the ability to apply it to anything. And that's like looking at, we're over a decade of data collection there. We have the ability certainly to do like really deep sequencing on the microbial communities. We can go in and take biopsies, we can take mucosal samples. So we have the capability to do all of those things, but it comes down to really, I mean, like funding. It's super expensive to do like the whole genome stuff. They've got like what I'm doing in this project I'm collaborating on, we're doing shallow sequencing, which is a very new thing. Basically, you take like fewer reads, but you still should be able to kind of extrapolate the same quality of data. But the other thing is like we're still not able to identify all of the microbes that are present because we have to compare those to reference genomes. So we can't really like look for who we don't know is there. So that kind of has to come first, probably, that we have to like be able to identify everyone that's there. I would say that's probably like 20 more years, maybe 10 if we assume that like science, improves exponentially.
Yeah.
And then we have to design intervention studies that would allow us to approach a causal relationship. So something better than like, the long-term observational things that we're doing. But we also have to, I think, be like, have a better, like more homogeneous way of doing these studies. Because like some labs are doing like RNA based, some are doing DNA based, and then like the whole mouse thing and like all different diets. So probably there's going to have to be like some consortium where a bunch of like the leading researchers in the field come together and say, this is how we're going to do this. Now we have all of this. We're able to identify everyone that's there. Now here's how to best design these studies. But it might not actually ever even get to that place because like, look at how, like the genetic nutrition thing completely pooped out because it was like, oh yeah, there's more to nutrition than just genes. There was like a reemergence of like eugenics and euthenics from like the early 1900s came back because people were like, hey, let's just use our genetic material to tell us what to eat. So I don't know that we will even necessarily ever get there in terms of like being able to determine what might be optimal. Mm-hmm. But we're probably within 10 or 20 years of saying we have a more concrete idea of like the keystone species, like what should we see in the human gastrointestinal tract, probably. Mm-hmm. And then a better idea about what dietary patterns might be most supportive of that. And then maybe from there we can start getting to, more refined use of probiotics, for example. Like probiotics are not complete garbage. Like they do seem to have applications. So that might be kind of the next thing to get into is like, okay, if we can't really do much with the whole microbiome, can we maybe look at better ways of using probiotics or better ways of like making antibiotics more specific, for example?
Mm-hmm. So this is a two-parter, and both please wear the scientist hat, but obviously you're quite the practitioner. And this is off topic, but what do you feel that a lot of your clients, people that you speak to in general about nutrition, put too much of a focus on when it comes to their diet and nutrition? And then the antithesis, things that they should focus on, but they tend to neglect.
I would say there are two perspectives there. Mm-hmm. When it comes to what they're doing, and then when it comes to why they're doing it. Those are the two biggest misconceptions or biggest, I think, like shifts of focus that need to occur. So certainly people are usually, in terms of just like what they're doing, they're putting a lot of emphasis, like maybe an overemphasis on worrying about like the really minute things, like what supplements do I have to take? Or, you know, like are artificial sweeteners, like, you know, really damaging? Or, you know, certain foods, like, can I eat this food? Can I not eat this food? Like, what happens if I eat too much of this one food? And like, not looking at the big rocks of, okay, first we have to look at energy balance, and then we can look at nutrient density. And then from there, do we even care about nutrient timing? Because you're not an athlete, you're a recreational exerciser, and that's just like a thing that's going to make things too complicated, you know, extra complicated. And then looking at, you know, the supplements. I mean, there are like 2 supplements that are probably helpful for people. It's caffeine and creatine, you know? And so, so they're kind of trying to nail, you know, put a bunch of nails in, and they haven't put the walls up yet. So that's in terms of like, you know, the very specific, like, what are they trying to do? And certainly when it comes to gut health, they're like, you know, kombucha and probiotics and all this stuff. And they want to know like what it is they can do to optimize their gut. And I'm like, it's like, how many vegetables do you eat in a week? You know, like, let's just start there. But I think probably the more foundational and influential and evocative change is the why they're doing it. Corn is actually a vegetable. You know what gets confusing though are avocados and tomatoes. Avocados are a fruit, and tomatoes can be a fruit or a vegetable depending on if you're horticultural or agricultural. So that's why I'm like, just plants. And yeah, like soda's okay. It's like they have these very rigid rules, you know, about what they think is allowed or not. And a lot of that comes down to just the misinformation in the media. But that really foundational shift is shifting from not just what is the right thing to do or what's the best thing to do, or like, I need more knowledge about X, Y, and Z, and like, why am I doing what I'm doing? So it has been shown empirically, putting calorie counts on a menu does not actually change people's caloric intake. It doesn't change their food choices. Now, certainly part of that is that they need some context for those calorie counts. You know, if you've never counted calories, you know how many calories you are supposed to eat, or you don't know what a calorie is or what it means. Seeing some numbers is just going to be like, okay, that's great. I have no idea how to apply that. But even when people have that knowledge and they can apply it, sometimes they will not do the thing that they think they want to do, because in the moment they actually want to do something different. So instead of eating like a macro tracked calorie counted meal, they eat something that's really enjoyable, or they eat because they're feeling emotional, or you know, they're stressed or they're bored. So they focus so much on what are the things that I need to change about my diet, and forego what is it that got me to this place in the first place. You know, like what has my lifestyle been up to this point, and then why am I doing those things? So even shifting from like, let's maybe not focus on all the things that you want that you think need to change right at this moment. You know, we can imagine like, what would your ideal day look like? Okay, cool. What's been your stumbling points to get there? Like, what are your obstacles? So for example, I worked with a client recently who said that she wanted to change the amount of bread she was eating. She was like, I eat too much bread. And I was like, oh, okay. You know, how do you know that it's too much? She's like, I don't feel good. I don't feel good when I eat that much bread. I think like maybe I have a problem with bread. And it was a gut health thing. Like maybe I have a problem with bread. Maybe it's gluten. I was like, okay, it could be. You know, let's talk about what happens when you're eating the bread. Well, it turns out that it wasn't just bread. It was that the root cause was that when she was going to go out to eat, because she only ate bread when she went to restaurants. So it was an off limits food that was super restricted. And then when she went out to eat, she would eat lots and lots and lots of bread, because she would kind of restrict earlier in the day. She'd be so hungry that it was hard for her to eat an intentional amount of bread that felt good. So she focused really on like, I want to change the amount of bread that I eat. I think like bread is the thing I need to change in my diet. And it really was, let's change the schedule leading up to you going out to eat, so that when you go out to eat, you're not famished. You can eat 1 piece of bread or 2 pieces of bread, stop when you're satisfied, and still have room for dinner. And lo and behold, that was it. It wasn't that bread was causing GI distress. It was the amount of bread was causing the GI distress. So that's been, I think, really pivotal for my clients in shifting that from being very like outcomes based, like changing, you know, let's just change your veggie intake, to kind of like foundations based. And so that's why I try to marry these so that, you know, when a person comes in wanting to like optimize their gut health, or they have concerns about their gut health, that there's an educational aspect to it, but it's also helping the client learn more about themselves and why they're doing the things they want to do or don't want to do.
Awesome. And thank you again, Dr. Fundaro, for speaking to the class this evening. I don't see any extra questions for them, so I guess technically three, but I know we didn't contextualize the fact that obviously also you've, you were pretty decent at the bench press back in. If you want to talk a little bit about how's your training going these days, anything you're planning on doing, and then I'll have one more hopefully fun question for you.
Oh yeah. You know, it's been really interesting in how COVID and lockdown have, I think, changed people's perspectives about the role of like exercise and training and sports in their lives. So I had, you know, I've been lifting since I was 19. So that is 14 years now. I don't mind telling people how old I am. It's been a long time. I really fell in love with it when I was in college. And yeah, I've done a fair few bench press competitions and powerlifting competitions, but I've dabbled in endurance sports, physique sports, you know, and really, I think have run the gamut from one end to the other in terms of like, you know, being a trail runner all the way to like powerlifting. And I found, you know, when I was faced with being like not having access to the gym or weights or anything like that, quite often I think people tie that to like all of their health seeking behaviors. So it's like, well, I can't train, then like I don't feel like, you know, eating in any way, you know, that's organized and whatnot, and they just like feel really bummy. And so people were like super stressed out and like lost this intrinsic part of themselves. So since that happened, I really have gotten into like, why am I moving my body and why am I eating the foods that I'm eating? And my goal became not like, oh, increase my strength or whatever, because I'm working with resistance bands or nothing. And it just got to like, I'm going to move in a way that feels great. And my intention for my meals was to feel better after I ate a meal. And when that's your goal, I think it's much easier to make intentional choices, because it's like, oh, I could eat 20 Reese's cups and I would feel really bad after that. But if I eat 2, I feel great. Or if I eat 3 or whatever. And so I kind of have changed the way that I think exercise fits into my life. But now that I have access to an in-home gym, which I feel very fortunate and grateful for, I've gotten more structure back into my training. And but still, the purpose really for me is just to maintain bone mineral density and muscle mass, and challenge myself and have fun and feel great. I don't really have any competitions or anything planned. I moved to Arizona a few months ago, and I've always loved hiking, but I was doing a lot more hiking until it was like 118 degrees, and then I was like, oh, maybe I'll take a break from that. It's a little hot. So yeah, I just feel that, you know, I feel better when I'm moving around, whether that's walking, hiking, lifting, doing yoga. So I engage in all those things because they're enjoyable. And when things open back up, I'll probably get into like dancing or something.
Awesome.
Mm-hmm.
Awesome. So last question.
Yeah.
It's okay to have no good answer to it, but what's something that you, when you do present, and I obviously, well, when we're not in a COVID world, you travel a lot for this and otherwise, what's that you wish more people would ask you about when you're presenting? Like, A, you find it interesting, or you feel like people are kind of forgetting, because obviously your presentations are there to hopefully answer their questions, but you wish some people would dig deeper into just because you're a fan of it.
Well, I would say, I mean, like, because I talk about the gut microbiome mostly when I'm traveling. That's like the number one thing that I'm booked for. And it is cool to talk about that. And so I would say gut microbiome specific would probably be, I wish that more people would kind of ask about the limitations. You know, I think they're very quick to assume that like, oh, we have some practical recommendations from this, right? You can tell me what to do. And I wish people were like more interested in knowing like why I can't tell them that yet, because maybe then they would be, you know, more satisfied with the presentation. But I think as a whole, I like to talk more about coaching and facilitating behavior change. And like, I got into education because I found it so fulfilling to empower people and to, you know, help them progress in their learning journey. And so I feel the same way about coaching. And so that's one thing that I'm like, I talk about that more on podcasts, I think, and more recently, and hopefully that continues to grow. But yeah, we're even kind of like marrying how we can inform people without invalidating their experience. Because when a person thinks they might have bad gut health because they're having some GI distress, it's important that we validate that their experience is real. I'm not going to say like, no, you feel fine, there's nothing wrong with your gut, but to also do that in a very client-centered way, to say like, I acknowledge that this is happening to you. Can I give you some information? And then can we grow from here to pursue an intervention that would be effective?
Awesome. Well, Dr. Fundaro, thank you again so much for your time. I greatly appreciate you speaking to the class this evening. I know a lot of them are quick with the mute button, so hopefully they say thank you in the chat. Hint, hint, kids. And, yeah, have a great evening.
You too. Thank you so much. And thank you guys for tuning in. Oh, awesome. You guys are so welcome. Oh, very cool. So yeah, like I said, please feel free to reach out. You can email me vitaminphd@gmail.com or vitaminphd on Instagram, and keep learning, you guys. You're awesome.
All right. Have a great evening, and I'm going to give them some more trouble. So thank you again.
Awesome. Very welcome. Bye.
Bye-bye. Let's talk about vitamins and minerals. So at the end of the day, there is a number of vitamins that we need in the diet. Now, we have our fat-soluble vitamins. So these are ones that are going to obviously go in a solution in fat. And then we have our water-soluble. Those are the ones that obviously are going to go in solution in water. Now, our fat-soluble are going to be vitamins A, D, E, and K. And then our water-soluble are going to be all of our B vitamins and then vitamin C. Now, I'm not going to insult your intelligence and read to you guys what each of these do. Instead, we're going to go ahead and talk about some that are more prevalent or with a higher likelihood of people being deficient in them, depending on things like dietary decisions, like are they vegetarians, are they vegans, are they eating too much or not enough of one nutrient or another. So for the most part, as long as you're eating a pretty good mixed diet, you're not going to have an issue with any or a lack of any of your vitamins and minerals for that point, except for it seems to be vitamin D and vitamin K are the two that when it comes to both vitamin and a mineral that you've got a greater likelihood of just being a little deficient in it. And especially when you're working with athletes, because you get a lot of magnesium from eating vegetables. And vitamin D, you get it from very limited food sources that aren't fortified, but that is something that your skin can naturally produce, but that requires getting enough sun exposure that you can take advantage of it. So, exercise is going to slightly increase our needs of some vitamins. It, however, is typically not going to be that large, and it is important to understand that we can test for things like blood levels of vitamin D, and we're going to touch briefly on the concept of what's known as triage theory, which is when the body doesn't get in enough of a given nutrient, it effectively has importance of what do we need to do today so we don't die? What do we need to do if we've got a lot more to make tomorrow better? And then if we got even more, so not getting into points of which we're talking about that tolerable upper level of intake. We're not getting close to toxicity, but we can effectively take care of everything we need. So vitamin K is a great example of it, where you're going to find that vitamin K is very important for blood clotting. However, it's also important for calcification of your bones. And it does so by actually obviously helping your body lay down calcium so it's not floating around the bloodstream, which in turn, theoretically, high levels of vitamin K intake in turn are going to decrease effectively the amount of calcification that's going on in your arteries. Now, that's that triage theory. It's really important right now that we don't die of blood loss.
it's pretty likely you're going to have a cut, you're going to have a bruise, you're going to have something, and it can also be internal hemorrhaging, that if you don't have the ability to coagulate, you're going to die. So it's not so important to make sure that we're not calcifying our arteries when it comes to surviving today. But eventually that can become a problem, obviously 30, 40, 50 years down the road when you're having a myocardial infarction or a stroke. So like anything else, you just want to make sure that you're getting enough. And then anytime that someone is starting to show symptomology of a lack of intake, that's when we're going to worry about potential supplementation. Now, one thing that's important right here is when we're talking about vitamin D actually is effectively, it's really functions more like a hormone. And notice it looks relatively similar to cholesterol, though there are some major differences. It's going to effectively convert into different forms. There's different forms of vitamin A, there's different forms of vitamin K, vitamin E itself is kind of a family. So the different types in turn are going to have different amounts of bioavailability, meaning how quickly or how difficultly it is going to be for our body to go ahead and be able to pick up those nutrients. So keep in mind, for the most part, if you're taking in enough calories, you typically are going to just accidentally get in enough of your vitamins and minerals. And what we're going to see when we're working with an athlete, in that it's not so if they fail on one day, you're going to have an issue. It's if they chronically are not taking in enough of the vitamin. So it's more about the average. So if one day you're 2 times above the RDA, the next day you're at 0, 2 times above 0, your average is going to be right at the RDA. You're probably going to be all right. But if you only have the one day where you're getting enough and the majority of the days you're not, that's where you're running yourself into an issue. And specifically when you're on a lower calorie diet, you're more likely to have issues with obviously not getting in enough of a given nutrient simply because you're not eating as many calories. And if you're not being very, very thoughtful about what is going to be the sources of those, you're going to have a problem. So we then have, when we talk about, I briefly brought up different versions of vitamin E. There are some different versions of vitamin K, which do have different effects on that, removing calcifications of the artery. Tocopherols and tocotrienols, those are going to be obviously other pieces of vitamin E, and it is completely okay if all of those words don't make sense. The key is, once again, it's a family of chemicals. So one of the problems when you supplement vitamin E, you're typically only taking in one form. You're not taking in all of the different potential forms of vitamin E, which are potentially going to have slightly different effects of antioxidant performance in the body. Now we then have minerals. Minerals are going to be into 2 different buckets. We've got macro minerals. This is going to be a mineral that we need getting closer to a gram of per day. And then we have our micro minerals, ones that we only need to take in a number of milligrams per day. So our macro minerals are listed a la here, along with what also should be listed is potassium, but potassium you rarely are going to have, it is, I was thinking about with a big K. Boy, that's special. I'm glad I recorded that so you got that to enjoy in the future. And to understand that we're losing a certain amount of all of our minerals when we're sweating. So notice it's not just that sodium and chloride, but we're also going to be losing a certain amount of calcium, magnesium, and potassium. So hence why you need to make sure that you're eating enough of all of those minerals throughout the days. And specifically, we need to think about, okay, going from triage theory, Hannah, what do you think is more important for calcium's use in the current, in the moment, so in the present, to keep your bones strong or to allow your muscles to function?
I would guess bones strong, since that's what we've always been taught.
So then if you think of it that way, your heart is a muscle. So it's okay if your heart fails as long as your bones won't break.
Yikes.
Yep. No, that's okay. That's okay. But does that make sense now? So
Yeah.
when it comes to that triage theory, your body prioritizes removing calcium from your bones so that you can have muscular contraction, so that your nervous system can fire. And because of that, if you're not getting enough calcium, getting back to going with athletes, now you're going to be leaching calcium from the bones slowly but surely to allow muscular contraction. And even you see it as part of your sport. Which of your athletes, which of your teammates are most likely to have a stress fracture from overuse?
Probably sprinters.
Who do you see in boots all the time on your team?
Oh, cross-country athlete, the long distance.
Bingo. They're not very heavy. The problem is that calcium is being obviously shuttled more towards their muscles so they can fire. Then they're able to take in enough calcium in their diet, along with vitamin D, vitamin K, and magnesium, and kind of their interactions there, in order to not just allow muscle function, but to not just maintain, but increase bone mineral density. So you've literally witnessed, gosh knows how many times with your own teammates, of the body making the priority of it's more important to avoid becoming something's meal than to risk having a hairline fracture of your bones. So, does that make sense?
Yeah.
Awesome. So, what we have here is just a simple effects of, what are good sources? Once again, eating a wide variety of food choices. If you decide to be a vegetarian, that's where calcium can be a little bit more difficult to get in, especially if you're, doing, shall we say, low rent vegetarianism, where you're just eating a lot of, cheesy puffs and bread. And you need to make sure you're getting lots of green vegetables. You're making sure that you're getting in some legumes, and obviously egg yolks also aren't an option. And that's also where you get a lot of the fat soluble vitamins also. Now, when it comes to your chlorine, your sodium, we were just talking table salt. That's pretty easy to make sure any athlete's getting enough of. And then obviously it's added to so many processed foods. It's pretty rare to meet somebody that's not getting enough salt in their diet. If anything, people tend to go the other way. But what's fascinating is a high sodium diet does not necessarily mean you're going to have high blood pressure. What you're going to find is most people, the amount of sodium in their diet doesn't really actually influence their blood pressure. A solid third of the population or more are salt sensitive. So a high salt diet will lead to high blood pressure, especially if they're sedentary. But what's fascinating is there's a very small percentage, less than 10, that they get paradoxical high blood pressure from low salt diets, low sodium diets. So it is important to understand how the body responds. Now, notice when we're looking at the options for magnesium, it's very easy to overlook it. And the easiest sources, hands down, are going to be eating a lot of nuts and vegetables. And it turns out a lot of people aren't really ingesting that. So hence magnesium is an easy one, especially for a hard training athlete, to be deficient in. Of all the minerals, that's the one that most humans that eat a normal mixed diet have the highest likelihood. Now, when we're looking at those microminerals, which ones over here do you think are most likely for an athlete to be deficient in that happens to be a vegetarian?
Ooh. You said vegetarian?
That is a vegetarian.
I don't know. Hold on. It looks like it would be, I don't know.
That's okay. That's okay. It's probably going to be iron.
Oh, okay. Yes.
Because iron, every single nutrient when it comes to your vitamins and minerals have a certain amount of bioavailability, meaning how well we digest it. Iron and heme iron, meaning you get it from an animal source, that has the highest amount of bioavailability. And your average person, of all the iron you ingest, you typically only actually pick up about 50% of it in the best of times. So your plant-based iron quite frequently has much lower bioavailability, along with you do it with or if you take it in with fiber, that decreases the absorption. If you take it in with vitamin C, it actually increases the absorption. There's a lot of things that are going to influence your body's ability to pick up a number of these different minerals. And if you're not getting in much in the first place and the body's not that efficient with it, it's very easy to find yourself in a situation where you don't have enough. Now, zinc and copper actually compete with each other. And you're going to find the same type of competition with calcium and magnesium to a certain extent. So your mineral intake, it is really important to realize that you need to make sure you're getting enough. And when people supplements like megadose with vitamin C, or sorry, with with zinc, they can actually potentially run the risk of not getting in enough copper. And copper is really important, not just for iron absorption, but also for the formation of red blood cells. So, taking enough. If you're an athlete, you probably need a little bit more than your average person, but if you're still eating a relatively balanced diet, you're good. At the end of the day, maybe supplement with something like a multivitamin. And we're talking, you could just get Flintstones. You don't really need to be that hardcore parkour. But the bigger issue, hands down, is always going to be when we happen to not be taking in enough each day. So if you're on a lower calorie diet because you're trying to make weight in a weight class, because you just have to be really small for your sport, it's very easy to find your way below the RDA, especially if you're doing a specialized diet where you cannot eat as much. And if we look in the bottom left, so you're looking at calcium, notice the people with the highest calorie intake are safely way above the RDA for calcium. Meanwhile, these athletes, and specifically, I believe these are the female athletes on the lower calorie diets, those are the ones that were below the RDA. Now, when it comes to calcium turnover, just remember your body is constantly using that calcium to lay down new bone, but also to allow us to have our normal function of our nervous system and our muscles. So anytime we need more calcium in the bloodstream so it can go to those tissues, it's being leached from the bone. Anytime we have an excess amount, along with we have vitamin D to help with the mineralization, and we've done that signaling, think back to biomechanics, Wolff's Law and everything, then we're going to increase that bone mineral density. So your bones, you actually have an ability to store calcium, so to speak. So you can have way higher bone mineral density than you need effectively. However, you still need to make sure that you're getting in enough on average throughout the week. And if you megadose a number of different vitamins, minerals, you don't necessarily always ingest and absorb it all. Now, our micronutrients, specifically vitamin E, vitamin C, and then selenium, are all very important for dealing with what's known as reactive oxidative species and reactive nitrogenous species. These are naturally produced due to high intensity exercise and otherwise. And the problem is when you produce these, in turn, you can really do a lot of damage to your cells, meaning damage to the membranes, the proteins, the DNA. So your body effectively wants to try to get rid of these antioxidants as quickly as possible. Sorry, also copper and zinc is important here. So using your vitamins, minerals, you produce things like, you probably have heard of glutathione before, which is the major antioxidant in the body. Obviously, we've talked both about vitamin E, vitamin C, and you're also going to have a number of different essentially flavonoids, so different types of chemical components of plants that are going to help with free radicals. But we, strangely enough, want to have a certain amount of free radical production because it's going to actually allow us to go and actually signal for positive changes in our muscles and our vasculature. Kind of like a little bit earlier, we talked about inflammation. You want inflammation in the areas you've trained in otherwise, so your body is going to go ahead and effectively mount a response that you're going to go ahead and adapt and improve. But you don't want there to be just crazy wanton and excess amount of inflammation, just like an excess amount of reactive oxidative and reactive nitrogenous species inside of the body, because that actually does cause cellular aging and cellular damage. Questions?
No, not that I can think of.
Awesome. So this is just another way of showing those chemical interactions of how you have productions of the reactive nitrogenous and oxidative species, how effectively you're getting this produced from a number of different inputs inside, or like as far as nutritional and environmental things you're exposed to, along with the actual work that your body is doing. So all of these are going to potentially be doing damage. So have you ever used hydrogen peroxide on a wound? Which I know is not the standard of care and is not something you should be doing a lot of.
I'm sorry, what'd you say? People walked in.
That's okay. Have you ever used hydrogen peroxide on a wound?
Yes. Isn't it just like bubbly water though?
It is bubbly water, but that bubbly water is causing really quick cell death of pretty much anything it's touching.
Oh, okay.
That's why if you pour it all over your skin, you see it bubble up and then it looks like your skin's kind of dried and flaked off.
Yeah.
Yeah, so you don't want to do a whole lot of that if you can help it, because go figure, that's too many reactive oxidative species, specifically hydrogen peroxide on that tissue. So you want to make sure you resolve that as quickly as possible. Now, obviously, when we think about the big uses of micronutrients, specifically iron, we're talking oxygen transport. Now, things like zinc, things like iron are also very important in your own immune system's functioning and are cofactors in a number of enzymatic processes in the body. So when we're talking about iron, and I'm sure you've had teammates that have had issues with being anemic, they're going to look at their hemoglobin levels. Now, what's also interesting to look at is going to be both what is known as their ferritin levels and their transferritin. Think of ferritin as your money in your bank account. Transferritin is the money in your be in your wallet, and then the actual blood hemoglobin levels is your actual money in your hand. So you're still just transferring one to the other. If you're broken, all three, you're screwed. You can have a lot of money in your hand, have nothing in your wallet, and have nothing in the bank. So that would be someone that is effectively, they've got no stores that they can draw from. You can have pretty much no money in your hand or not much there, but if you still got a filled wallet and a lot of money in the bank, you're going to be okay because you can always withdraw funds. But iron is something that we can store slightly. And obviously, if we keep going away and decaying or those stores because we're not replenishing them with enough iron in the diet, we can have some issues. In fact, getting back to those athletes when they convert over to a vegetarian and vegan diet, and don't get me wrong, I don't expect you to get it right, but I'm very interested. So if we figure an athlete, we're just making a cliche female long distance runner. So you have the monthly phlebotomy schedule that is menstruation, and you have got the excessive red blood cell death from the foot strike of walking. If they were to switch over to, let's say, a hardcore vegan diet, and they are not supplementing with iron, they're not timing their iron, they are just doing a very poor job of getting iron in their diet, how quickly would you suspect they would start to present with issues of anemia?
Ooh, I mean, it would be like obviously within a month because, right? Because they would start like their period and stuff. Like that's like a lot of blood loss, right?
Well, obviously it depends on the woman. So that's one big point. But it depends on obviously how good the stores are. So if you had somebody that didn't have good stores in the first place, you're right. At the end of a full calendar month, they could really start to see a hit in their performance. If you're well topped off and you obviously then are doing a very poor job of getting an iron, you could last as much as say about 3 months before you'd start to present.
Oh, I mean, that kind of makes sense because doesn't it take like about 120 days anyways for blood cells to like,
They usually live about 90 days. So,
okay.
but the simple reality of it is you have these athletes that can convert over to a hardcore, like I said, vegan vegetarian diet, and then they're not keeping track of their iron. And, maybe there's a little placebo effect. Maybe they do feel better because they're getting a lot more fiber in their diet. They're getting a lot more of micronutrients in different ways. They're getting more magnesium definitely in their diet. And so they're like, oh my God, I feel so much better since I converted over to this diet. And then after 2 weeks, they still feel pretty good. After 3 weeks, they're feeling good, but maybe a little bit wonkier. And then they're slowly getting to the point where they're showing that deficiency. So that's one of the things to be careful with if you have an athlete that converts over aggressively to a diet where they might then lend themselves to issues of not taking enough micronutrients, not just obviously iron, but obviously all of your other vitamins and minerals, that it's not like if you're screwing up, you're going to feel it in a day. You're going to have some of these effects that come on in only, unless you're doing electrolytes. If you're really screwing that up, you might only make it a day and feel like garbage. But when it comes to a lot of your vitamins, minerals, from vitamin C, obviously to iron to vitamin D, you can have pretty low intakes and not feel any negative effects, not to present with scurvy. until it's, or rickets, until it's literally been months of inadequate intake. Does that make sense?
Yeah, that makes sense.
Awesome. So when it comes to cofactors, and specifically we're talking about effectively our B vitamins, this is their world. They are incorporated at one point or another into not just our wonderful, well, because it actually is important for NAD, notice biotin itself, for helping ourselves convert into oxaloacetate and pyruvate, along with they're going to be important in intermediates in the citric acid cycle and the electron transport chain. That's also another location where iron is important. So you're going to find pretty much all of these vitamins and minerals in certain extents in different foods. Obviously, when it comes to sodium, going with more effectively salted products are immediately going to have more. Notice beef itself plain actually is a relatively low sodium food, but how many people that you don't know season their steaks? Because that person is a monster if they don't. But meanwhile, you can have things that you wouldn't necessarily think about, like baked beans, having, you know, pretty decent amount of sodium per serving. It makes sense how you'd find a decent amount in pretzels, but then certain things like spaghetti sauce can actually have, I've gotten soy sauce is pretty much just straight salt. You can take in a huge amount of it and not even realize what you're doing. And since this is Kentucky, I do have to point out gravy powder. Now, on the other side, when it comes to potassium intake, now I understand that coconuts aren't here, but notice a banana is. But out of all of the foods listed, which of them is actually the highest in potassium if you really wanted to have a high potassium intake, Hannah?
Which foods, left or right?
Sorry, potassium. Notice.
Oh, yes, got it. Well, it looks like potatoes.
Yeah, absolutely. So the funny thing is potatoes are actually even higher, but of course they don't have as good of a lobbying of talking about how much potassium is in it compared to a banana and otherwise. So yeah, and it turns out potatoes are a lot less expensive than coconut water, though bananas are actually pretty inexpensive themselves. That's not really an expensive fruit to buy. So when it comes to looking at micronutrient data, this is going to be from a number of older studies where each of these numbers is an individual sport. So you've got the actual intake of the average athlete, followed by the extra line on top is going to effectively be, excuse me, I believe that is going to stand for supplementation. Now notice for the most part, on average, your athletes were taking enough vitamin C, B vitamins, but there were, in these, I believe group 6 was bodybuilders, and then group 12 was, there was some type of endurance sport. I think it was some type of long distance, or not long distance, but a little bit more of a short distance, where they literally, you do have risks of athletes not getting enough of their RDAs. And to the RDA of vitamin C, you pretty much just need to eat 1 orange. That's it. It's not a lot, but obviously if you're not eating a lot of fruits and vegetables in your diet, you can run yourself into some issues. Now, when we're looking at our micronutrient intake, it's important, once again, how bioavailable this is. Are we taking this in with other minerals? So is there anything else that's competing with it? Other vitamins, that doesn't really compete as much, but this is all done in context with iron. Fiber and phytates are both going to, and tannins are all going to decrease your ability to absorb it, whereas you're going to increase the ability to break it in or break it down and absorb it, not break it down, it's already iron, which is going to be through vitamin C. Now, heme iron, which you get from beef, is the most bioavailable. Ferrous oxide is literally rust, and that's what's ground up and put into a lot of iron supplements, which is garbage. But that's really what people are sold because they don't know any better. And then plant-based iron, unless you're talking specifically from a certain type of soybean, is more bound up obviously in the plants. And so you're going to have more of an issue with extracting it and having that full absorption. Now, once you get that iron into your bloodstream, there's a number of tissues that are going to use it. It can sometimes be bound to some plasma proteins. You're going to obviously urinate a certain amount of it out and sweat a certain amount of it out, along with, yes, depending on your natural phlebotomy schedule that you have with your body or potential GI issues. So this is just for iron. You're going to see the same type of situation here with sodium and chloride. You really don't have any of those issues with the uptake so much, but like I mentioned before, we've got some competition between zinc and copper, and you're going to still have limitations on how much you're taking. Actually, yeah, calcium is not going to help that either. Steak and an orange if you really want to help your iron absorption. So it can definitely potentially help, especially when we're getting into more electrolytes and making sure that we're not having issues with not taking in enough of our minerals each day. But at the same time, it's just important to talk to each athlete, figure out what's going on, figure out what are their normal dietary decisions and choices, and then from there try to get a good working understanding of where the greatest likelihood is of them being inadequate. So at the end of the day, just eat a balanced diet. Take a multivitamin if you maybe really want to, and otherwise you tend to be good. If you are a hardworking athlete that is sweating a lot, you're going to have a much more massive demand of all of your minerals because you're sweating them out, especially when we're talking about sodium, potassium, and chloride. But don't worry too much there about being inadequate as long as you're eating enough food and you're eating whole foods. And don't be afraid to salt your food if you're that aforementioned athlete that is out there working hard and sweating. So we're going to, whenever we meet up next week, we're going to talk in groups about different bioavailable forms of magnesium, which we're talking about oxide, which is garbage and is a decent laxative if you're not moving regularly. Then you've got Trax, you've got glycinate, you've got, oh gosh, the one that's theoretically more bioavailable for the brain. I can't think of the name of it right now. It's not theanine, not theanine. Hopefully it comes to me later. But there's a lot of different things that go into it. Selenium is important whenever we're talking about things like potentially being, it's a positive antioxidant, which is also implicated with that entire concept of the triage theory. Vitamin D, because it effectively works as a hormone, is important not just for bone mineral density, but also for muscle function. And it seems to actually maybe even be tied a little bit in mood, and a lot of people happen to be deficient in its intake. And then vitamin A when it comes to the different isoforms and how that's important for visual health. So, and vitamin K, we already talked about, and there's MK7 and a couple iterations, but that was quick. What questions you got for me, Hannah?
Why are people always so deficient in vitamin D?
So vitamin D is something that you're typically only getting it in fortified foods, or if you're eating a lot of whole fish, like I hope you like cans of sardines, and whole eggs. And then on top of that, it's really a hormone that you're meant to get from sun exposure. And since most people are living a life indoors and they're not getting out in the sun a whole lot, and when they are out in the sun, they're not having a lot of skin exposure, they're much more likely to be deficient. In fact, Division I athletes, specifically indoor athletes like basketball players, volleyball players, and otherwise, unless they're obviously playing beach, they have pretty decent risks of being deficient or at least non-optimal when it comes to levels of vitamin D in their body.
Okay, interesting. Because I mean, like as a thrower, I spend, at least just the fall and spring outdoors, but I do spend a lot of my, obviously in the winter, inside throwing because of indoor. So.
Bingo. And then don't forget about the fact that since it is important in adaptations to training, you want to make sure you're never deficient. And on top of that, it's not like you're running around throwing that stuff in a two-piece swimsuit.
That's true. That's like the sprinter's outfit.
True, but even then, they're not outside for that much each day. The cross-country girls are in pretty minimal amounts of clothing, but that's only until it starts getting legitimately cold. And then same thing, you're not getting as much sun exposure.
Yeah, that's true.
And that's only for people that are nice and pale like you and me. Folks that are much more darker complected, they need a lot more sun exposure for the same amount of vitamin D synthesis. And there's genetic components to your own body's natural production of vitamin D thanks to sun exposure.
Okay, interesting. Also, doesn't like skin color play a role in that? Like, depending on how like darkly pigmented you are, that's like the less exposure you need in the sun.
No, more.
Oh, more. My bad. I got that mixed up.
No, it's okay. That's why I touched on that with the pigmentation, where yes, exactly. The more like Casper the Friendly Ghost you are, the less sun exposure you need for that vitamin D production. But obviously, the less time it takes for you to get a sunburn, and then the antithesis. So if you're really going to it would be your African-American basketball players and indoor volleyball players that would probably be at your greatest likelihood. So, yeah. Any other questions, comments, concerns?
No, I think I'm good.
All right. Well, thanks for sticking around. Sorry for boring you, but have yourself a great Thursday, and I will talk with you more soon.
All right.
Okay. Have a great one, Hannah. Bye-bye.
You too. Goodbye.